Today, my friends and I are talking about cellular respiration by Paige, Ella, Taylor, and Kira. So first, we're going to begin with the big picture. What is cellular respiration? Cellular respiration is the process of breaking down a substance, usually glucose, into simpler products. This happens using a series of chemical reactions producing primarily the end outcome of ATP. To the right is a visual image showing the inputs and outputs as well as the main steps of glycolysis, step 1, and we'll talk about the rest as we continue throughout the presentation. There are 4 steps which I will briefly touch on in the next slide. The steps in cellular respiration. First, we have glycolysis, which only occurs in the cytosol because all required material is located here. There are 2 phases that take place, which are energy requiring and energy releasing. Step 2 is the prep reaction. Pyruvate turns into acetyl-CoA, which is the key to the next door in cellular respiration. That door is the Krebs cycle. Step 3. It initiates the oxidation of the intermediates, and it is the step in between to get the products to begin in the next step of cellular respiration, which is the ETC, the electron transport chain. The ETC is the formation of a proton gradient, separating the matrix and the intermembrane. The main elements in this chain is the moving of the H from a higher concentration to a lower concentration in the matrix. This is a chart showing where it happens. So to start off, glycolysis occurs in the cytoplasm. 2 ADP go in, which is labeled here, 1 glucose, or glucose in general, and 2 NAD+. What comes out, then the 2 NADH, which is the coenzyme, 2 ATP molecules, 2 pyruvate. The net ATP is net gain of 2, and it's the first step. At this point, if there's no oxygen, it will perform an alternative pathway, which we will talk about later. The second step is the prep reaction or pyruvate oxidation. It occurs in the mitochondrial matrix. There are 2 NAD+, which goes in, and 2 pyruvates. The coming out is the NADH, the acetyl-CoA, which is the coenzyme molecule that attaches to an enzyme's active site. It also helps the catalyze reaction. 0 net ATP, and it is also the second step, and CO2 comes out as another byproduct being pulled off. The third step is the Krebs cycle or the citric acid cycle. This happens in the mitochondrial matrix. There are 2 ADP that go in, 6 NAD+, 2 FAD+, and 2 acetyl-CoA. The coming out is the 6 NADH, 2 FADH2, and CO2 is also pulled off here. The net ATP is 2, and it's produced by the substrate level phosphorylation. The other notes is it is this third step. The end is the glucose, which is fully broken down. A little trick to remember here is oil rig, which is oxidation is lost and reduction is gained. And the concluding step, step 4, is the oxidative phosphorylation. It happens along the cristae, which is the inner membrane of the mitochondria. It is the inner membrane of the matrix. And what goes in? 32 to 34 ADP, 10 NADH, 2 FADH2, and 6 oxygen. What comes out is 10 NAD+, 2 FAD+, 32 to 34 ATP, and 6 H2O. The net ATP is 32 to 34, and it is the concluding step, step 4. It has multiple steps in this phase within the electron transport chain and chemiosmosis. Go back really quick. I have a door question here. So what are the order of the 4 steps in cellular respiration? That's right. Glycolysis, prep reaction, Krebs cycle, and oxidative phosphorylation. And remember, each step has an output that is the key to unlocking the start of the next step. I'm going to pass this over to my friend Kira.
We have some additional vocab. Autotroph are producers that make their own food, which is photosynthesis. Example, plants, algae, and cyanobacteria. A heterotroph are consumers that get food from eating other plants or animals. Examples are animals, fungi, and bacteria. Substrate level phosphorylation, the process in which a phosphate group is added to ADP to produce ATP. This occurs in a glycolysis and Krebs cycle. Mitochondrial structure, a double membrane energy harvesting folded inner membrane to increase SA to volume ratio. So we have the first step, which is a glycolysis. I first have a door question. Where does glycolysis occur? It occurs in the cytoplasm, which is outside the mitochondria. Glycolysis is known for sugar splitting. It partially oxidizes glucose, 6C, to carbon pyruvate, which is a 3-carbon chain. It has a net gain of 2 ATP, 2 NADH. It also makes 2 H2O. PFK is an enzyme that speeds up glycolysis. Its inputs are glucose, a 6-carbon molecule, 2 ATP, and 2 NAD+. Outputs are 2 pyruvate, which is a 3-carbon molecule, 4 ATP, net gain of 2 ATP, and 2 NAD+. So going a little more in depth with the picture, glucose is put in, and then 2 pyruvate, 2 ATP are put in to produce ATP. This and then creates fructose biphosphate, then splits into DHAP and GP3. GP3 is oxidized, NAD plus is reduced to NADH, and then energy is required above the line and then is released below the line. And then 2 GP3 and 2 ATP are produced through substrate level phosphorylation and then turned into 3PG and then turned into 2PG. H2O is then released and PEP is made. PEP donates a P to ADP to produce 2 more ATP through substrate level phosphorylation. 2 pyruvate are made through this process. This is just another way of looking at it. So it says that step 1, which is above the line, is energy investment. Invest 2 ATP, glucose is phosphorylated, rearranged, and then split into 2 G3P molecules. Step 2 is energy payoff. It harvests 4 ATP and 2 NADH. G3P is oxidized and then broken down into an intermediate molecule, which is PEP, and the PEP donates a P to NADP to make ATP and 2 pyruvate. Next, I'm pass it on to Paige.
Okay, the prep reaction. So this occurs after glycolysis and before the Krebs cycle. So what did we end with in glycolysis to begin the next step in cellular respiration? That's correct, 2 pyruvate. So the prep reaction happens only in the presence of oxygen. It occurs in the mitochondrial matrix. So pyruvate moves from the cytoplasm to the mitochondria by active transport with the help of pyruvate translocase, a transport protein. In this prep reaction, NAD plus gets reduced to NADH, and 2 pyruvate molecules formed from glycolysis join with CoA enzyme and produce the 2-acetyl-CoA. The end results, the 2 pyruvate, which is 3-carbon molecule, turn into 2-acetyl-CoA, which is a 2-carbon molecule, which releases 2 carbon dioxide molecules and 2 NADH. Next is the citric acid or Krebs cycle. So this occurs in the mitochondrial matrix. The purpose is to break down glucose and produce carbon dioxide. The net gain is 2 ATP, which is produced by substrate level phosphorylation, 6 NADH, 2 FADH2, and those are both electron carriers, which will later be used. And then acetyl-CoA, which is 2-carbon, combines with oxyl acetate, which is a 4-carbon, it's right here, which forms citric acid, which is a 6-carbon molecule. Then citric acid goes through a series of oxidation reactions, in which it loses carbon atoms to form carbon dioxide. Once it loses a carbon, hydrogen is available to reduce NAD plus to NADH. So you can see here the NAD plus becoming NADH. And then this oxaloacetate right here is regenerated to continue the cycle. And then this entire cycle is catalyzed by enzymes. And since you go in with 2-acetyl-CoA, this cycle happens twice. So next, I'll pass it to Ella to talk about oxidative phosphorylation.
Oxidative phosphorylation is the long way of saying that it's the process that creates energy through a series of proteins which line the inner membrane of the mitochondria. So in oxidative phosphorylation, there are 2 steps. We have the electron transport chain and we have chemiosmosis. The electron transport chain occurs where again? That's right, it occurs along the inner membrane of the mitochondria. It uses up 10 NADH and 2 FADH2. It pumps out H plus into the inner membrane space to create a proton gradient, which we'll use in the next step. NADH is converted into NAD+. What's that called again? That's right, that's oxidization. So the NADH is oxidized to create NAD+. The electrons then flow through. More electrons are added from the FADH2. They continue through until they're added at O2 in the end. All while that's happening, H+ is being pumped through the channel proteins to create a concentration gradient with a high H+ concentration here and a low one down here. The concentration gradient created in the electron transport chain is then used during chemiosmosis. In chemiosmosis, the H+ that were on the top go through this protein over here. What's the protein called again? That's right, it's called ATP synthase. ATP synthase spins when the when the H+ is going through it. As it spins, ADP and is added to a P to create ATP. What's that called again? That's right, that's called substrate level phosphorylation. At the end of chemiosmosis, there's a production of 32 to 34 ATP. I'm gonna pass it back. Oh wait, hold on.